It will also give you still pretty good. Yeah. Yeah, so it's generalized to backward working even though it's a change of I'm after this after it'll work so it's just
Okay, so I think we should get started. So we have the final set of talks for this afternoon and for the whole workshop, and we'll start with Gily doing this about apps.
Yeah, so I'm Gily Ginosar, and besides being Shiri's sister, I am a neuroscientist, and I will tell you about work I did in the Ulanovsky lab at the Weizmann Institute in Israel about action-specific spatial coding in flying bats. So we can do very different actions at the same space, and you would think that we might want a representation, a brain representation of space that would be different for this, to account for these different actions, for how we use space differently. So namely a joint representation of space and action. But that's not what we find the brain does. Space in the brain is encoded in the hippocampus and the entorhinal cortex. This is a human brain. You can see the hippocampus and entorhinal cortex. Now, in terms of anatomy and information flow, the medial entorhinal cortex acts as a bottleneck relay station between hippocampus sending and receiving information to the whole of the neocortex. So the medial entorhinal cortex is really the relay station. And focusing on that, I'll show you a few spatially tuned cells that we find in the entorhinal cortex. There are border cells that fire at the border of arenas. There are head direction cells that fire at a specific head azimuth of the animal. There are speed cells. There are object vector cells that fire next to all objects in an arena. And there are grid cells. Now, here is a beautiful example of a grid cell. It was recorded in the Moser's lab. And it's a top view of a box where a rat is running around. And in black, you can see all the places where a single grid cell fired in space. And so obviously the most prominent property of this pattern is the hexagonal symmetry, and that's what grid cells are well known for. And it's also a symmetry that you can see in many places around nature, especially when you are trying to tile surfaces. And almost most of what we know about grid cells comes from rodents running on surfaces. And in my previous project, I asked, what does the grid pattern look like in 3D? And for that, we turned to bats and let them fly in volumetric space. So we let bats fly in this 5 by 6 by 3 meter room, flew around, and they were allowed to land on landing platforms. And while they were flying, we were recording wirelessly from their entorhinal cortex. Now, when we go from 2D to 3D, we considered various possibilities of what we might find, and varying in how ordered they are. But our best guess was that when you go from 2D hexagonal lattice arrangement, we would find 3D hexagonal lattice arrangement. So let's look at the data. This is a firing rate map of a single cell in the flight room, and it's in the form of a heat map. And you can see that there are multiple firing blobs, multiple firing fields. And here are 8 different cells in the same room. And ultimately, what we found is that these fields are not arranged in a hexagonal lattice arrangement or in any other lattice arrangement. Instead, they were ordered in a local order. They exhibited a local order where neighboring fields had a fixed distance on average between them, but no hexagonal symmetry. So to sum this up, grid cells in 2D exhibit a global hexagonal symmetry, in 3D only a local order. Now, together with Jonathan Al-Khadef, Yoram Burak, and Chaim Sompolinsky, we wanted to find a phenomenological model that would capture in 3D this local order, but no global order. So we took inspiration from many physical systems that have multiple particles that could range from being more or less ordered depending on the parameter regime. So so we chose to model it using pairwise interactions, treating the fields as though they were particles. So this means close by fields would repel each other, far away fields would attract each other, and this is simulations. Using one model, we were able to capture both the hexagonal grid in 2D and this local non-global order in 3D. And apparently, we shouldn't really be surprised by this result because the same thing happens in hard spheres as well. This is a physics paper. They are packing spheres, and in 3D, the arrangement just gets stuck in this local minimum solution and it doesn't crystallize. But in 2D, it arrives to the global minimum solution of hexagonal lattice very quickly. Now, regardless if we're talking about 2D or 3D, what we know about the brain and grid cells and other cells in the brain, that they have an objective representation of space, regardless of what it is we do or the actions we take. But now I want to show you some new results. So this is data that we collected in the grid cell data set. So it's the same setup. The bats are flying around in the room, and they're allowed to land on these platforms. And they can rest on them, and the platforms are also pseudo-randomly baited with food. And what we find is that there are cells in the entorhinal cortex that have a firing field next to one of these platforms. Each time it's a different platform. It's not object vector cells. First of all, because they don't fire next to all of the platforms. And more importantly, because when we look carefully at the data that creates this firing field, and we divide it into the flights of landing, takeoff, and just passively flying by the same location. This is the vicinity of the platform. We see in gray, you can see the trajectories, and in red, you can see the positions of the spikes. So we see that this firing field was made predominantly from landing. Now, this actually creates almost action-specific spatial map, because now here I take all the data, gray is trajectories and red is spikes, and I make a firing rate map. But here I take only the data from the action by position. So only in this case, only the data from landing to platform 6, and I get this map. But now I get all the other data whenever you weren't landing on platform 6, and I get a different map. So action-specific spatial map. This is not objective space. There it was landing, but we also get takeoff. Here's another example where a cell is showing, you can see here, it's firing next to platform 3, but only in takeoff, not in landing, and not when it's passively flying by the same location. Here is another example where you have two preferred platforms, both in landing, and you can see different maps. And also you can see that this signal is kind of overlaid in this example on other firing. Now, why are we seeing this now? Why are we finding this only now? Experimentally, we record spatially tuned cells from animals that are doing a random walk. They are just randomly running around boxes searching for pellets of food. And this is by design because we want them to sample space uniformly. And so in a way, we only know how the brain represents space under a random walk. But here with bats, naturally, they have these two directed, distinct, well-defined, purposeful actions of landing and takeoff. It just happens. And so I think this is why we see it now in the bats. Now, most of the cells that we found, this is a very wasteful, in a way, wasteful code. Most of the cells we found represented one action and one position. So you can see on the left the number of significant platforms on the x-axis, mostly around 1. And it seems here that the number of significant platforms doesn't increase with the number of overall platforms, although we only checked between 6 and 11 possible platforms. So I don't know what will happen if I'll have 100 platforms. Would it be like a percentile, or would it be still 1 or 2? Or Pietro, like yours, will it be like 1 or 2 and many? I don't know. Interestingly, we see that there is a comparable number of signals between landing and takeoff. And this makes us think that it's probably not a predictive reward signal like we would think if it was just landing, because you are predicting the landing is rewarding. It's the food is there, you can rest, and so on. But there's takeoff as well. Now, the entorhinal cortex, its superficial layers send projections to the hippocampus. Superficial layers is where we find object vector cells. Sends projections to the hippocampus, and the hippocampus sends projections back to the deep layers of the entorhinal cortex. And this is where we find our action position cells. So we think maybe this localized signal comes about from, has to do something with the fact that we have place cell hippocampus coming in to these cells. So localized information, bringing about localized information. The selectivity can't be explained by classical head direction tuning. So these cells fire in a specific head direction, because if I'm now firing only when I'm landing on this platform, then it's always one head direction. However, whenever the bat is in other areas of the room at the same head direction, it doesn't fire. You can see it here in the in black is all the behaviors or the instances it was facing the same direction, but not next to the platform. And then it doesn't fire, which is what you would expect from head direction cells. So it's probably not a classic head direction tuning. That can't explain it. It's also not a speed tuning. So you could say, maybe, we know that there is speed signal in the entorhinal cortex. Maybe landing or taking off from platform X has a different speed than taking off from other platforms. But that doesn't seem to be the case. The speed to the significant platform is pretty much the same, or has small differences from the one of all the other platforms. All right, so, okay, 2. Kim Stackenfeld and others' previous works suggested that grid cells may serve as a low-dimensional decomposition of spatial maps, hippocampal spatial maps under random walk. And I'm happy to talk about this offline, but I just want to mention work we're now doing with Daniel McNamee from the Champollion, and he's going to be here in the second workshop, where we're doing the same thing only for directed movement. And he sees that he gets elevated firing in the positions of actions under directed movement, at the position where action is possible, the position where the symmetry breaks, a little bit like our cells. So what I showed you is entorhinal cortex neurons that code for position by action, in a way giving us action-specific spatial maps, which is not how we thought the brain codes for space until now. And you could ask two questions. First of all, is it connected to valence? Because these positions are not any positions. These are important positions. The reward is there, and so on. The answer is probably yes. I don't know how to decouple it from experimentally, but these are actionable positions. These are positions where you can do an action. So it's probably connected to that. The second question, and I'm happy to hear your thoughts about this, why do we get this redundant representation? Why would we have a representation of, one position with one action? Why not have a landing cell or takeoff cell and a position cell and kind of activate them together? Happy to hear your thoughts. What we think is something along the lines of, it is wasteful in terms of number of neurons, but perhaps if we want to do a rapid shift between different action maps, then we want, we want to act in short delta t's. So we don't really have time to integrate two signals from pure neurons. So perhaps if you want to move quickly, you want all the information to be in one conjunctive neuron. But happy to hear your thoughts about this because we don't really know. That's it. I want to thank all the collaborators on these two works and my advisor, Nachonovsky, and you.
Time for maybe 2 quick questions. And so, Chandra, if you can set up what we do.
I think, so I mentioned this to you yesterday, but I'll just note here and then ask you a question. So what we see when we train recurrent neural networks to predict the next sensory input in a simulated environment is the emergence of exactly this kind of encoding. They come to encode spatial locations contingent upon. And if you think about it, for sensory prediction, that makes a lot of sense. What am I going to see next? Well, I have to know not only where I am, but what action I'm taking to get there.
Right.
Because that's going to affect the actual sensory perception.
Right.
And so I wonder if you instead gave the animal a landing space where there were many different directions it could land, you would actually see it be not just a landing specific encoding, but actually a direction / landing specific encoding, because that's what
because of the different visual.
Yeah, visual, or I guess in the bat's case.
No, they see.
Okay, yeah.
They see well.
Yeah, whatever, the different sensory experience it would get. And have you guys explored that? Because here it looked like there was one landing path. So it was always going to kind of get the same sensory inputs.
Right. Right. We looked okay, so 2 things. We looked to see whether, like in the pass by, if we have, behavior that is with the same head direction as the tuned head direction. And that's how by looking at frames in the pass by at the same position, that's how we said, okay, it's probably not head direction by position. But
predicting the future. So
right.
It's not going to bother predicting.
I see what you're saying. So we don't have this. The problem with this experiment is that it was, it's the data from the grid cells. So we didn't put the reason we had the platforms, we put it at the edges of the room so that we would have a lot of space, a lot of empty space. Yes. So this is a missing link in this experiment. exactly that. That would be interesting. Yeah, cool.
Okay, I think we should move on. So let's thank Gily.